Good morning, dear friends. The type we are going to study today is very interesting because it is one of the well-developed algal type and the most developed one among chlorophyceae, and the type is Chara. The Chara is a common plant usually seen in the stagnant water, in ponds, aquariums, tanks, etc. Some are grown in slowly running water. Chara baltica is grown in brackish water, and Chara fragilis grows in hot springs. Usually, Chara is seen in hard, fresh water where large amount of organic matter is present and which is less in dissolved oxygen content. They grow luxuriantly in the water containing calcium, and the surface of Chara is encrusted with some calcium deposits, so they are also known as stonewoods. In appearance, they are similar to the equisetum, so they are also known as aquatic horse tails. The equisetum is a pteridophyte, commonly known as horse tails. Since the Chara is aquatic, it is known as aquatic horsetail. The thallus of the Chara has a smell of onion due to the presence of sulfur compounds in the thallus. So it is used as an identifying feature of the genus in the community. Chara hathei grow trailing on the surface of the soil or substratum, and most of them grow up to 30 centimeters in length. Some are up to 1 meter height, and this hathei grows up to 2 to 3 centimeters long. The common Indian species are Chara ceylanica, Chara brownie, Chara gracilis, Chara hathei, Chara gymnoptyi, etc. As I said, it is a well-developed thallus, green in color, and has some calcium deposits. Coming to the structure of the thallus is differentiated into the green photosynthetic portion and the rhizoids, which are used for the attachment of the plant. The plant is much branched, multicellular, and macroscopic. These are the pictures and videos on the thallus organization and habit of Chara. You can see the main axis. On the axis, we can see some projections known as stipules. The axis is differentiated into nodes and internodes. In the nodal region, we can see two types of branches. These branches are known as branches of limited growth and branches of unlimited growth. The branches of limited growth are also known as the primary laterals. The branches of unlimited growth are similar to the main axis. They grow indefinitely and sometimes come back or grows downwards to the soil or substratum and produce rhizoids, growing as a separate plant and spreading the thallus. The stipules are developed on the basal nodes. So the thallus is much developed. We will see the structure in detail. And these are the reproductive structures, the globule and nucleus. The globule is the male reproductive structure or the antheridium, and the female reproductive structure is known as the nucleus. You will see the structure of globule and nucleus when we discuss the sexual reproduction. And this is a diagrammatic representation of the thallus where we can see the rhizoids, main axis, two types of branches of unlimited growth and limited growth. So now we will see the structures in detail. Coming to the rhizoids, rhizoids are elongated, narrow, uniseriate, multicellular structures growing from the base. They arise from the rhizoidal plates formed at the base of the main axis or from the peripheral cells of the main axis. These rhizoids are characterized by the presence of oblique septa. The tips of these rhizoids possess minute solid particles known as statoliths. The rhizoids show apical growth and they help in the attachment of the plant to the substratum, which is mud or sand at the bottom of the water body. They also helps in the absorption of minerals and nutrients from the substratum and also involved in the vegetative multiplication of the plant by the formation of bulbils and protonema. Coming to the main axis is long, branched, erect and differentiated into nodes and internodes. The internode consists of much elongated, uniseriate single cells. But these internodal cells are surrounded by some cortical cells in some species. If the internodal cell is surrounded by the cortical cells, such species are called as corticated species. If such cortical cells are absent, they are called as ecorticated species. The node consists of a pair of cells surrounded by many peripheral cells, usually 6 to 20 in number. This central cell and peripheral cells arise from a single nodal initial cell. These cortical cells are unique in origin. These cells actually originate from the nodal cells, the peripheral cells of the nodal region. These peripheral cells grows towards the internode. From the lower node, some peripheral cells grows upwards along the internodal cell. Similarly, some of the peripheral cells of the upper node grows downwards. The peripheral cells of the lower node growing upward are known as ascending cells or ascending filaments, and the cells which grow downward from the upper node are known as descending filaments. These ascending filaments and descending filaments meet at the center of the internode. So actually, these cortical cells are not a part of the internode, but actually the nodal cells which grows upward or downward and ensheath the single internodal cell. So this is a unique structure in Chara, and it is the beginning of layers of cells, and it is the beginning of the tissue structure, where we have seen the filamentous algae up to Idagonium, where the thallus is uniseriate multicellular filaments, but we can see some layers. These layers originate in a different way from the higher plants, but it is the beginning of this tissue system. Here in the picture you can see the arrangement of internodal cells and nodal region and the development of these cortical cells on the surface of internodal cells. On the surface of this main axis there are different types of appendages. Appendages are extensions or structures present on the main axis. They are the branches of limited growth, branches of unlimited growth, and the stipules. The branches of limited growth are branches arising from each node, and the peripheral cells gives rise to these branches of limited growth. They grow up to 6 to 20 cells. They arise in whorls of 6 to 20 based on the number of peripheral cells present, and are of limited growth up to 5 to 15 nodes in different species. They do not grow beyond that. Since their growth is limited, they are known as the branches of limited growth. They are also known as the primary laterals. They are also called as branchlets, branches of first order, etc. The stipuloids and reproductive structures are produced from the nodes of these primary laterals. But the branches of unlimited growth are similar to the main axis. They also arise from the peripheral cells of the nodal region, but they grow indefinitely. They also produce the primary laterals, sometimes grow downwards and reach the surface of the substratum or soil and produce rhizoids, gets separated from the mother plant and grows a separate individual. So they're exactly similar as the main axis. They bear the primary laterals. So they are also called as auxiliary branches or long laterals. Then the stipuloids. Stipuloids arise from the basal nodes of the primary laterals. They are short, oval, pointed outgrowths and are unicellular. In most of the species of Chara, like Chara burmanica, the stipuloids at each node is double the number of primary laterals. So they are known as bistipulate. If the number of primary laterals and the number of stipules are equal, they are known as unistipulate. When these stipules arise only in one whorl, the condition is known as haplostiphanous. And if two whorls of stipules are developed, the condition is known as diplostiphanous. Here you can see the haplostiphanous and diplostiphanous conditions in the thallus. Coming to the cell structure, we can see two different types of cells in the thallus. Those cells of the nodes are different from the cells of the internode. The nodal cells are isodiametric, smaller in size, and contain dense cytoplasm. They are uninucleate and contain few small ellipsoidal chloroplasts. The central vacuole is not developed, but numerous small vacuoles are present. The cytoplasm can be differentiated into an outer exoplasm and an inner endoplasm. But the internodal cells are much elongated, contain a large central vacuole. The cells are multinucleate and contain many discoid chloroplasts. The cytoplasm is differentiated into an outer ectoplasm and an endoplasm. The inner endoplasm shows streaming movements known as the cyclosis. The cyclosis in Chara is well studied for the last 2 centuries, and the mechanism of this cyclosis is well explained, and you can refer some of the papers I have added in the description. The cell walls of both these internodal cells and nodal cells are porous and ensure the continuity of cytoplasm. In the figure you can see the structural organization of the internodal cell and a nodal cell. So we have seen the habit and structural organization of Chara thallus. It is well developed when compared to other types we have studied in Chlorophyceae, and it is believed that from this Chara the higher plants evolved. It is the beginning of the multicellular nature of the thallus and well-developed chloroplasts and different types of cells on the same thallus. So it is much developed, and in the size also it is comparable to a higher plant, except that the plant body is a thallus. Next we will see the reproduction. The Chara reproduce by vegetative methods and sexual methods. Vegetative reproduction is by the formation of bulbils, amylum stars, amorphous bulbils, and secondary protonema. Bulbils arise from the rhizoids or from the lower nodes of the main axis. They are oval or spherical tuber-like structures, and upon detachment they develop into a new plant. There is a unique structure for the reproduction in Chara known as the amylum stars. These amylum stars are star-shaped structures developed from the lower nodes of the main axis. These structures contain specialized stars known as amylum stars, and so they are called as amylum stars. They are multicellular star-shaped aggregates of cells. They detach from the axis and grow into a new Chara thallus. The amorphous bulbils are irregular shaped groups of cells developed from the lower nodes of the main axis or from the rhizoids. They store large amount of reserve food materials, and they function as perinating structures. They develop towards the end of favorable conditions and remain detached from the plant during unfavorable season and regenerate when the favorable conditions returns. So they are perinating structures. The secondary protonema is filamentous or tubular structure developing from the rhizoids or from the primary protonema and develop into the thallus. just like the primary protonemia. So these are the vegetative reproductive structures developed in Chara. Coming to the sexual reproduction, the sexual reproduction is complex and unique due to the presence of the reproductive structures known as the globule and nucleus. We will study the sexual reproduction in the next class. Thank you.